Welcome to Mr. Chalk's Revision Tips. In this video, we will look at
mutations, beneficial mutations, and types of mutation. So, genetic variation is the difference in DNA among individuals or differences between populations. There are multiple sources of genetic variation, including mutation and genetic recombination. So mutations can occur spontaneously, often during DNA replication, but the rate of mutation is increased by mutagens. A mutagen is a chemical, physical, or biological agent that causes mutations. The loss of a purine base or a pyrimidine base often occurs spontaneously. The absence of this base can lead to the insertion of an incorrect base through a complementary base pairing during DNA replication. Free radicals, which are oxidizing agents, can affect the structure of nucleotides and also disrupt base pairing during DNA replication. Antioxidants, such as vitamins A, C, and E, are known as anticarcinogens because of their ability to negate the effects of free radicals. Beneficial mutations include things like lactose tolerance, rich colour vision, and in some a resistance to certain diseases. Beneficial mutations can confer an advantage on an organism possessing them and over time, so these mutations can spread throughout a population. So the ability to digest lactose, so the sugar present in milk, is thought to be a result of a relatively recent mutation. So the majority of mammals in the world become lactose intolerant after they cease to suckle. So the ability to digest lactose is found primarily in European populations who are more likely to farm cattle. So the ability to drink milk and process lactose as an adult helps prevent diseases such as osteoporosis. So this could also have prevented individuals with mutations from starving during famines. This mutation appears to have risen spontaneously more than once and has also been found in people in East Africa. A gene mutation is a permanent alteration to the DNA sequence that makes up a gene, such as the sequence differs from what is found in most people. Mutations range in size, so they can affect anywhere from a single DNA building block, so a base pair, to a large segment of a chromosome. So a gene mutation, like we've just said, is a permanent alteration, and there are some different ways that they can occur. So a substitution of a single nucleotide changes the codon in which it occurs. If the new codon codes for a different amino acid, this will lead to a change in the primary structure of the protein. The nature of the genetic code may mean, however, that the new codon still codes for the same amino acid, leading to no change in the protein that is synthesized. The insertion or deletion of nucleotides leads to what's called a frame shift mutation. So the triple code means that the sequence bases are transcribed consecutively in non-overlapping groups of 3. So this is the reading frame of the sequence of bases. So each group of 3 bases corresponds for one amino acid. So if there's an insertion, that means that all of the codons will eventually end up being altered. In addition to deletion of a nucleotide, move or shifts the reading frame of a sequence basis, this will change every successive codon from the point of mutation, right? So this isn't a frame shift. So the same effect is seen, however, many nucleotides are added or deleted unless the number of nucleotides changed is in multiples of 3. Gene mutations occur in single genes or sections of DNA, whereas chromosomal mutations affect a whole chromosome or a number of chromosomes within a cell. They can be caused by mutagens and normally occur during meiosis. A gene mutation, so the mutations can be silent but often lead to developmental difficulties. So changes in chromosome structure include inversion, which is a section of the chromosome breaks off and is reversed and joins back to the chromosome. Deletion, so a section of the chromosome breaks off and is lost within the cell. Translocation, so a section of the chromosome breaks off and joins to another non-homologous chromosome. And duplication, so sections get duplicated on a single chromosome.
Thanks for watching.